All right, thank you. So we've already had a very nice introduction on the virtual brain from Victor this morning, afternoon, I think for you. And Jennifer showed us a very nice example of these large-scale dynamic models. So I don't think I need to go too much into kind of some of the details of the neural masses and how they're linked together. But I'll just remind you that the population activity from these neural masses can be, I guess, expressed in our preferred neuroimaging signals. So this is, again, Victor's referring to the link, the direct link between source signals and your neuroimaging signals at different scales. But what I wanted to do today was specifically present the macaque branch of the virtual brain. And I was hoping to motivate parts of the discussion on ways in which we can actually refine these dynamic network models, especially in the macaque because of all the empirical data that we have. But we already had a very lively discussion before the break on some of the examples in which we can start to extend these models and refine them based on all the data that we already have. So what I will instead do then is give you an example of how we've already done that to the virtual brain macaque branch. And I'll remind you also that typically we use for the connectome combination of anatomical and diffusion MRI data to define the structural constraints of the model. So it would, we have in the connectome a set of weights which describe the fiber track capacities for the connection between all the regions in our parcellation, and also then a matching track lengths matrix. And both the weights and the track lengths together, as Jennifer has already mentioned, really constrain the dynamics or define the dynamics in the models. And then we can also refine these models using any individual's functional MRI data, for example, if that's the empirical data level that we're measuring. So we can do kind of some parameter fitting with a person's functional MRI. Now, what we've done to kind of take advantage of existing macaque data to refine the macaque model in TVB is use track tracer data as a set of priors. And really this was motivated by how tracer data is actually quite accurate, whereas diffusion data tends sometimes not to be diffusion MRI data. And Victor already showed you in the mouse, for example, how those tracer connectomes have greater predictive power of the functional connectivity, for example, in the mouse, and that's also the case in the macaque, as opposed to those individual diffusion connectomes. And so, although, especially in the macaque now in recent years, we've been able to acquire diffusion weight imaging data at incredibly high resolutions and produce these really nice whole brain estimations of structural connectivity, there are still disadvantages to those tractography connectomes. And one is that they can be inaccurate, and the second is that they can be susceptible to particular choices in tractography algorithms and parameters. And I'll show you a nice demonstration of this in this particular paper from 2017, where they had a set of ground truth white matter fiber tracks that they were then reconstructed with various types of algorithms in tractography. And you can see that depending on the algorithm and some of the choices made on how the tractography is done, you're going to get very different results and they differ quite a bit from the ground truth. In some cases, you get a lot of missing tracks. So the first number here is the percentage of overlap with the ground truth. And so, for example, this really poor reconstruction only reconstructed 26% of the original fibers. And in some cases, you actually get a lot of false positives. And so a lot of, so this second value is called, it was the overreach. So you get a lot more streamlines or tracks than were actually present in the original. And so given that, and given that in the macaque especially, we have this really nice set of tracer data to work with, we took the best of both worlds approach to, we took the tracer data as a set of priors. And what we assumed was that the tracer data is accurate. We borrowed from the tracer data in information about the directionality of connections, and because we know that that directionality is important, even at, let's say, the scale, even at the scale of neural imaging signals and functional MRI, we have shown how, you know, resting state networks and its organization can depend on whether connections are bidirectional or asymmetric. And again, if you remember back to the mouse data that Victor showed, he showed one example where if you symmetrize the mouse connectome, you get slightly less predictive power than if you don't. And so that, again, is some evidence that we should be using the directionality data that we have available from tracer connectomes. And we also borrowed from the tracer data, actually, a whole cortex parcellation from existing tracer data in the Macaque. What we did then for the virtual brain Macaque connectome is that from a set of tractography data from diffusion MRI and a set of 9 macaques, we estimated the weights of the connections, so the fiber track capacities and also the track lengths. And so I'll walk you through very briefly today kind of the workflow in which and how we built the connectome for the TVB macaque. And in the first step, we took these tracer priors and in a very particular subset of connections where we had information about the weights from tracer data, and we optimized our tractography settings based on those subsets of connections. So we tried to really, for each individual subject, get the best possible estimate from the tractography and the diffusion MRI data. We applied that optimal those sets of optimal tractography parameters to the whole cortex to derive an entire set of weights and track lengths for the entire cortex of the macaque brain. I'll show you then what's available, what's made available for the TVB macaque package, and then I'll walk you through an example of a resting state fMRI simulation. So this has already been mentioned today, and this is from Henry Kennedy's lab, and from in particular described in a paper from Markov et al., a really nice set of data where the weights have been provided in this tracer data for a subset of connections in the cortex. And so what we did was take the edge complete connectivity matrix, which includes 29 of the 91 cortical ROIs, and we tried to get our diffusion tractography to best estimate this, to get it to best match this tracer matrix. And so the way we did that is by systematically varying the curvature threshold and tractography distance correction, whether we applied it or not, and the percentage of connections that we discarded at the end. And so I'm showing here, this essentially is the optimization target, that 29, that subset of 29 by 29 ROIs. And one example, monkey, where we varied along the rows here, the tractography parameters, and also along the columns, the percentage of connections that we discarded. And just the heat map here shows the area under the ROC curve. So essentially a goodness of fit of those tractography parameters. And C here in panel C is the best fit set of tractography parameters. And you can see that it actually does a fairly good job of recreating that tracer matrix. And then in other tractography parameters, you actually get some highly varying results. So I'll just show you here in total for all 9 animals averaged the tractography matrix and what it looks like as compared to the tracer matrix. And then just comparing the weights, or so the fiber track capacities of the tractography estimates against the ones that were provided by Markov and colleagues along the x-axis here, does a pretty good job with a correlation coefficient of 0.7, which is fairly high. And so now that we have a good set of tractography parameters for every animal, we then reran the tractography using a whole cortex parcellation, and to try to estimate now the connectivity across the whole brain or the whole cortex. And so for every animal, we have a set of weights and a set of track lengths for the whole cortex. And what we did was average across 9 animals to get our TVB macaque connectome. And so what we did was keep the tracer weights, the tracer edges. So we assumed the tracer connections to be true, the ground truth. And what we did was apply the average tractography weights and lengths to each of those connections. And you can see, just to give you a sense of how good those weights are, it follows a log normal distribution. So in red here are the tractography weights distribution, and it follows a log normal distribution, kind of very similar to what the Kennedy lab has described in tracer data. Okay. And the track lengths that we achieved were as expected, so they increased with Euclidean distance. They were correlated with Euclidean distance, and also, intra-hemispheric connections were on average shorter than interhemispheric connections. Okay, so this connectome is what's built into the TVB package currently. What's also available, and what Jennifer has also mentioned is possible, is our cortical surface files for cortical surface level modeling and simulations. If you wanted a kind of a finer spatial grain than regional and then a regional simulation. And so in the last part, I wanted to show you the example resting state FMRI simulation that we did on this, and how we can achieve by doing this kind of refinement of the model using existing empirical data, we can achieve a fairly reasonable simulation of FMRI resting state networks. Okay, so the constraints on this are that Macaque connectome in the TVB package. Each region in this case was represented with a neural mass model, and specifically the reduced Wang model. And it's all coupled together based on these weights and track lengths. And then the population activity for each ROI was simulated, and then a human dynamic response function was applied to generate bold responses. And then we computed functional connectivity using Pearson correlations of the time series for the simulated data, and then did a parameter optimization with the empirical functional connectivity matrices from empirical data collected from those same macaques in resting state fMRI. Okay. And so this parameter optimization was done by varying systematically both the global coupling parameter, that so kind of the gain of of how coupled all the regions are together, and also sigma here, which is a noise parameter of Gaussian noise that's injected into the excitatory population. So here on the left is that fitting of the global coupling G in the rows, and the noise parameter in the columns. And so the heat map shows you the goodness of fit as determined in cosine similarity between the simulated functional connectivity and the empirical functional connectivity. The asterisk shows the maximal cosine similarity. So this is the best fit model parameter set. And so panel B here shows the functional connectivity for that particular simulation as compared to the average empirical functional connectivity. And when we, what we did was do a principal components analysis extraction of the time series, and the simulated time series, to see if we can essentially reduce or simulate known resting state networks. And that PCA produced 3 dominant, or more than 3 components, but the first 3 components of that PCA were a frontal network, a superior temporal network, and a visual network that looked and corresponded to what has been shown in resting state FMRI studies in the macaque. Right. So I'll just end here by sharing what is up next for kind of the macaque TVB branch, and also actually kind of more generally the non-human primate branch. One is the TVB macaque stroke model. So we have a set of macaque data in both neuroimaging and Utah array recordings following stroke or lesions in macaque. What I'm really excited about also is the virtual marmoset brain. And so we have plans to do essentially the same procedure in creating a marmoset connectivity connectome for the virtual brain by applying a set of priors. And in this case, again, the priors come from tracer data from the Marmoset Brain Connectivity Atlas that's been made available. So some subset of these connections create an edge complete matrix that we can use to kind of try to fine tune our tractography of the whole marmoset cortex. And of course, you know, we've already, in part of the discussions that we had earlier in the before the break, you know, we have had kind of behind the scenes discussions very briefly about the exciting new receptor atlas that's available now in Macaque, and also, you know, the heterogeneity of excitatory and inhibitory populations across the cortex. And so I really liked how Alan put it, so where, you know, if the data exists, it can be incorporated, and I'm hoping that we can kind of just continue that discussion later in the workshop. So I'll end by thanking all my collaborators, Randy, of course, our collaborators at University of Western Ontario who provided both the Marmoset and the Macaque data, and other members of the TVB group. Thank you. I'll end there.